Good morning, everyone. This is Shriya Samant, Assistant Professor, IIMT College of Pharmacy, Greater Noida. Today, the topic of my video lecture is plant tissue culture. So, moving on to the first slide, what plant tissue culture is. So, plant tissue culture is basically a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a culture medium of known composition. It is widely used to produce clones of a plant in a method known as micropropagation. Plant tissue culture was a new addition to the methods of plant breeding that developed around the 1950s. It makes the use of a part of plant to generate multiple copies of the plant in a very short duration. So like it's displaying in the image. So basically we take a tissue sample, we scrap it from the parent plant. Parent plant means from which we are producing the further clones. Then this particular tissue sample has been placed into the agar medium, which consists growth medium, which consists of nutrients and different growth hormones which are required for the growth and development of the plant. After that, after few time, samples develop into the tiny plantlets, which then further are planted into the compost. So basically, in short, plant tissue culture means with the help of plant's tissue, we culture it in a lab. So next is, what are the advantages of plant tissue culture? Okay, so the first advantage of plant tissue culture is that the production of genetically modified plants it causes. So, with the help of plant tissue culture, we can produce genetically modified plants. Means genetically modified plant or GMO, genetically modified organism, means with the help of plant tissue culture, with the help of genetic engineering, we will be planting the required or desired genes in the plant. Next advantage is the plantlets are obtained in a very short time with a small amount of plant tissue. The new plant produced are disease free. The plant can be grown throughout the year, irrespective of the season. A large space is not required to grow the plants by the plant tissue culture. The production of new varieties in the marketplace speeds up. This technique is being used for the production of ornamental plants such as dahlia, chrysanthemum, orchids, etc. Production of exact copies of plants, and last is crop improvement. So these are the different advantages of plant tissue culture, of using plants tissue culture to produce different copies or clones of the plant. Moving on to the next slide, different types of culture. So these are the different type of culture. Culture means the medium in which we will be growing. So these are the following different types of culture as per the requirement. First, it is flower organ culture. So, flower organ culture, as the name suggests, the flower buds or the mature flowers are collected, then they are washed thoroughly and dipped in 5% teapot solution for 10 minutes. Again, rewash and then transfer them into laminar airflow cabinet, which again, surface sterilization is done by immersing it in 5% of sodium hypochlorite, washed with distilled water, and using the sterilized faucet, we transfer it to the flower bud on the mature flower to culture tube containing the media. Now we'll incubate the culture for about 16 hours to 24 degree at 24 degrees Celsius under the light. So this is how we do the flower organ culture. Next is anther and pollen culture. So the names, the name suggests, we take the anther or the pollen part to produce the clones. This is a technique by which immature pollens is made to divide and grow into tissue, either callus or embryonic tissues, primarily to produce haploids. Haploids means the plants with N chromosomes number, known to be the anther culture. Haploid production through anther culture has been referred to as androgenesis. Next is shoot tip culture. As the name suggests, we'll take the shoot, we'll excise shoot tips from plant species, then we'll transfer it into a nutrient medium containing growth hormones, and after some days, roots will start developing and it will develop into a whole new plant. Next is root culture, in which the roots are excised from the plants, sterilized, and transferred to a fresh medium. The lateral roots continue to grow and provide several roots maintained in the continuous culture developed into a whole plant after few days. Next is protoplast culture. Isolated intact protoplasts undergo cell wall regeneration followed by rapid cell division and formation of small cell colonies. The cell colonies are subcultured to a new agar medium to form a callus or to a liquid medium for the establishment of the cell suspension cultures. Next is embryo culture. Embryo culture is a component of in vitro fertilization wherein resultant embryos are allowed to grow for some time in an artificial medium. Next is your ovule culture. So, experimental system by which ovules are aseptically isolated from the ovary and are grown aseptically on chemically defined nutrient medium uh, under control condition. Last is your leaf culture. Leaf culture, in the leaf culture, the excised young leaf primordia or immature young leaf of the shoot apex in a chemically defined medium when they grow and follow the developmental sequences under the control condition. Moving on to the next slide. So in this, we can show how, in a pictorial way, how we do the culture. So for example, here we are taking the tissue sample from the leaf. Tissue sample from any region of an adult plant is cultured. Then it has been transferred to a culture media which will contain agar, nutrient medium, and growth hormones. After some days, undifferentiated callus will be formed. Callus means basically a cell production will be there. Callus will be separated and single cells will be cultured, and these single cells will further generates new plant. Next is nutritional requirement of plant tissue culture. Means what are nutritional things we need to grow the plant. So first important nutritional culture medium is to maintain the vital functions of a culture. The culture media consists various elements. They are follows. So first important nutritional requirement is inorganic nutrient which have macro and micronutrients. Next is organic nutrients like vitamins, carbon, energy source, growth regulators. Next is solidifying agent, antibiotics, and at last it is pH. So talking about macronutrients, which comes under the inorganic nutrients, macronutrients include six major elements, which are nitrogen, calcium, magnesium, phosphorus, potassium, sulfur, etc. These elements, as the name suggests, are used in large amount as a stock solution for plant growth and development. Next is micronutrients. These elements are required in a very small amount or in a trace for plant growth and development. Example, iodine, copper, cobalt, boron, and zinc. Next is organic nutrient. Vitamins like thiamine, vitamin B1, pyridoxine, vitamin B6, nicotinic acid, vitamin B3. Next is carbon or energy source, basically glucose, sucrose, fructose, maltose. And last is growth regulators such as plant hormones which are required to promote the growth of the cells like auxins, cytokinins, jubilins, etc. Next is solidifying agent, which is again used for preparing the medium, which it is used to improve the oxygen supply and support the culture growth. Solid media are generally preferred compared to the liquid media. For this purpose, substance with strong gelling capacity added into the liquid medium, for example, agar or alginate, gelatin, silica gel, etc. Next is antibiotics, streptomycin, kanamycin, etc. These are the antibiotics used so as to prevent the growth of microorganisms. Last is pH. The pH of a nutritional medium is generally adjusted between 5 to 6. Next is general procedures involved in plant tissue culture. So these are the different steps which we conduct while conducting or while doing or before doing the plant tissue culture. So first and foremost thing is sterilization of glasswares. Whatever glasswares we'll be using in the lab for performing the plant tissue culture, all those particular glassware should be completely sterilized, means completely free from microorganisms so that it does not deteriorate the plant tissue culture or it does not deteriorate the procedures. Next is preparation of sterilization of explant. Explant means the piece of plant which we have excised and with which we will develop further clones. Third is production of callus. Callus means a soft tissue that forms over a wound or a cut plant surface leading to healing from explant. Next is proliferation. Proliferation means rapid increase in number of something. So after this, we'll do the proliferation of cultured callus. Next is subculturing of callus. That callus will be further subcultured. And last is culture suspension culture. So if we talk about sterilization of glassware, like I said earlier, All the glassware used in the tissue culture are firstly washed and sterilized properly. And to make them free from any dirt or waxy material or bacteria, all the glassware should be kept overnight dipped in a sodium dichromate solution. Next day, the glassware should be washed with fresh tap water and then with distilled water. For sterilization, glasswares are placed in hot air oven at 120 degrees Celsius for around an hour. For preparation of sterilization of explant, for the surface sterilization of explant, chromic acid, mercury chloride, calcium hypochlorite, sodium hypochlorite, or alcohol are used. If you talk about the production of callus from explant, callus is an unorganized, undifferentiated cell mass. In this step, the sterilized explant is transferred aseptically onto the nutrient medium contained in a flask. Now the flask is transferred to a BOD. BOD means biochemical oxygen demand incubator for maintenance of culture. BOD is used in measuring the amount of oxygen consumed by microorganisms in water or wastewater samples. Then sufficient amount of callus is produced within around 8 days of incubation. In this, the temperature is adjusted to about 25 degrees centigrade plus minus, and some amount of light is necessary for callus production. Next step is proliferation of callus. Proliferation, as I told before, rapid increase in the number of something is called proliferation. So if callus is well developed, it should be cut into small pieces and then transferred to another fresh medium containing some amount of hormones which supports the growth. The medium used for the production of more amount of callus is called as proliferation medium. Next is subculturing of callus. After sufficient growth of callus, it should be periodically transferred to a fresh medium to maintain the viability of the cell. And this subculturing will be done in an interval of 4 to 6 days. Suspension culture, it contains a uniform suspension of separate cells in a liquid medium. For the preparation of suspension culture, callus is transferred to a liquid medium which is agitated continuously to keep the cell separate. Agitation can be achieved by rotary shaker system attached with an incubator at 50 to 150 RPM. So another pictorial description of process of plant tissue culture. So first of all, we take the parent plant. Parent plant means from which we'll excise the explant, and which that explant will be used to grow the further clones. After parent plant, we'll do the selection of the explant from the mother plant. Then inoculation will be done. Light and warmth is given for shoot initiation. Callus will be produced. Further shoot will be formed. These shoots formed will be transferred to rooting media, which consists of higher oxygen concentration. And at last, the plants are kept for hardening in greenhouse or sold to a nursery and then potted up. So this is how we do the plant tissue culture procedure. Next is application of plant tissue cultures, means where we can apply or what all applications or in which all field plant tissue culture is used. Tissue culture has given a mechanism for propagating and genetically improving commercially valuable plants. Tissue culture has been used to study characteristics of growth, metabolism, reproduction, physiology, and nutritional necessities of the plant under controlled circumstances. Next application is in vitro hybridization, and embryos which are normally do not survive inside seeds can be developed into tissue culture to produce new plant, and it can be used for interspecific hybridization. The production of multiple of plants and the lack of seeds are essential pollinators to produce seeds. These are the different application of the plant tissue culture. Moving on to the next slide is edible vaccine. Edible vaccine, as the name suggests, edible means what we can consume through oral cavity. We can basically whatever is eatable is edible. So edible vaccines are the genetically modified crops that contain antigens for a specific disease. The edible vaccines offer many benefits over traditional vaccines due to their lower manufacturing cost and the lack of negative side effects. Transgenic plants are used as vaccine production system. These are the mucosal targeted vaccines which cause stimulation of both systemic and mucosal immune response. Transgenic plants are the plants that have been genetically engineered using recombinant DNA technology, which is transgenes, in order to introduce desired genetic traits novel to the original plant. For example, Bt cotton, or Bt means Bacillus thuringiensis. It is a group of soil microbes that are used to control certain insect pests. So Bt cotton is a genetically modified pest-resistant plant cotton variety that produces an insecticide to combat the bollworm. Bollworm is a worm which uh, destroys the cotton plant production. So, with the help of plant tissue culture, with the help of genetic engineering, such variety of cotton is being produced, which has an insecticide in their genes. Next, edible vaccines are subunit vaccines which are selected genes are introduced in where the selected genes are introduced into the plant, and the transgenic plants is then induced to manufacture the encoded protein. Initially, it was useful only on preventing infectious disease, but now it has also found application in preventing of autoimmune disease, cancer therapies, etc. Edible vaccine is also known as plant-based vaccine. The concept of edible vaccine was developed by Charles Artson in 1990. So, edible vaccines may offer many benefits over traditional vaccines due to the lower manufacturing cost and the lack of negative side effects. They are is easy to administer, they are easy to store, readily acceptable delivery system for different age group patients, yet cost effective. So this is how we prepare an edible vaccine. So whatever desired genes we want, we take that genes from the pathogen. Cloning of the desired gene is in vector. Plant transformation vector, which further produce the delivery of the gene in desired crops. These transgenic crops with desired antigen like tomato, cucumber, carrot, lettuce, etc. Orally, when we'll take these vegetables which contain that desired antigen, the we will have that protection from the pathogen by eating these plants, by eating these vaccines. Next, advantages of edible vaccines. Ease of administration is the first and most important advantage of edible vaccines. You can easily administer it orally. There's no need of medical personnel, avoidance of sterile injections. It is economical in mass production and transportation, well easy to store, heat stable, eliminating the need of refrigeration. Growing plant is much cheaper than producing vaccine. Plant cannot host most human pathogens, so the vaccine will not pose a danger to humans. So these are the different advantages of edible vaccines. So along with advantages, there are few limitations as well for edible vaccines. So first limitation is that the selection of best plant is difficult. It takes time. Second limitation is evaluation of dosage requirement is difficult because if you're eating the vaccine in a form of vegetable, you don't know how much do you require. So the dosage requirement is pretty difficult. Third is certain foods like potato cannot be eaten raw, and cooking the food might weaken the medicine present in it. So example, if we have produced a potato which has edible vaccine in it, so obviously we have to cook it because we cannot eat it raw. So while the process of cooking could weaken the medicine present in it. Another limitation, another drawback we could say. Plant are living organisms that change, so the continuity of the vaccine production is might not be guaranteed. The edible vaccine could be mistaken for regular fruits and could be consumed in large amount, then might be safe. And if the vaccines were grown in field or on trees, the security would be a big issue. So these are the different limitations for plant tissue culture. And that's all for the slide. Thank you so much.